All right, good afternoon, everyone. So it is Saturday, the 24th of April. So we're going to go ahead and start. Now, again, today is our very last session as far as the recorded lectures are concerned. I also sent out your study guides for basically all of your remaining exams, both lecture and lab. So that means you do have your lecture exam number 4 coming up on this uh, approaching Saturday, and also your lab exam. Actually, not your lab practical. So it's going to be only your lecture exam number 4, and then your final lab assignment that you would be turning in on genetics and human development. And then the Saturday after May the 8th is when we have our comprehensive exam and also your lab practical number 4. So all of those details have been sent out along with the study guide. And please make sure you're using it to the best of your advantage. If any questions come up, please feel free to let me know. Also, final reminder for your case studies, as well as your see what we have here. Right, so the lecture and lab study guides, as I mentioned, they've been sent out. Reminders for your case studies. Please make sure you're able to turn those in before we hit the deadline. And also your lecture quizzes, which are on McGraw-Hill Connect website. There were some questions about those. So there's like 7 of those quizzes, and they need to be turned in before the deadline, as was mentioned as well. Finally, your lab work, that is due tonight, by 12 midnight. And I've already sent out the final assignment for next week as well. So that pretty much completes all of the things that we had on the calendar. If any other questions, feedback, concerns, suggestions come up, please feel free to let me know. All right, so let's go ahead and start. So here we are on slide number 178 on chapter 28. So we have basically talked about the human reproductive system, both in terms of males and females. And as we appreciated that there are more similarities in the two than differences. So they're pretty much different expressions of the of the same type of physiology and anatomy as we saw. So what we are looking at here is two terms, your genetic sex as compared to the phenotypic sex. All right, so what is the difference between the two? So as far as the genetic sex of an individual is concerned, that boils down to his or her chromosomes, right? So when you look at human chromosomes, again, we talked about how we have 23 pairs in an individual of those chromosomes, one from mom, one from dad. And the first 22 pairs are exactly quite alike. They're called the homologous pair. The two chromosomes are similar in size and shape. So those are called autosomes, okay? And they carry traits like hair color, eye color, skin color, height, weight, all those kinds of phenotypic expressions. But the last pair, which is your pair number 23, there you have two possibilities. As you can see here, you can have an XX configuration where the two are very similar again, homologous chromosomes, and that makes you a genetic female, or you can have an X and a much smaller Y chromosome next to it, and that makes you a genetic male. So what is the phenotypic sex then? If that is what genetic determination of sex is, well, just because you have an XX or an XY does not always equate to you physically expressing as male or female again. And we are not even getting into gender at this point. Gender is, or gender identity is how an individual feels or perceives himself or herself or to be, right? So that is the social and cultural and psychological aspects of being male or female, or neither, or both. So, but in pure genetic terms, genetic sex is determined by your last pair. So phenotypic sex has to do with the appearance of your internal and external genitalia, as we see here. So typically, if you have ovaries as your gonads, reproductive glands, then you're considered female phenotypically, while if you have testes, then you are considered a phenotypic male. And this distinction becomes quite apparent by the 7th week of development, as we can see here. All right, so then moving on. So what determines sex, biological sex, is something called the SRY gene in males. All right, so the SRY gene, which basically stands for the sex determining region of the Y chromosome. That's what it stands for, the SRY. And this is only found on the Y chromosome, which you inherit from the dad. So if you do inherit the Y chromosome from dad along with the SRY gene, then you are genetically a male. And that's what we are looking at here, okay? So what this gene does is, during the first few weeks of development, it stimulates your body cells to secrete androgens, okay? Androgens are male sex hormones like testosterone, and it also sensitizes your cells to the effects of testosterone. So you develop male external genitalia, a male phenotype, and that's what we are looking at here. If the Y chromosome is absent, in other words, you have inherited X chromosome from dad as well, in other words, you don't have the SRY gene, then you continue on as a female, as a genetic female is what we are seeing here. All right, so next few slides talk about the primordial structures which turn either male or female during the early weeks of development in an individual, okay? So you're looking at what are called genital ridges. So if we move down here, all right, so if you look at this picture, right? So this is showing us the difference in development as far as the primordial structure is concerned, okay? So let's see if we can. All right, so I graduated to a different laptop, so I'm still figuring out the new system here. Okay, so what you're seeing here are what are called the primordial genital ridges, this area in the pelvic region, right? So if you do inherit the SRY gene on the Y chromosome from dad, then you start developing male structures here. So as you can see, the two testes, right, which will eventually become testicles when they descend down into the scrotum. You can see the vas deferens here, right? The epididymis, all of those structures developing along with time. Whereas in the absence of the SRY gene, the same primordial genital ridges turn into female structures. Here you see fallopian tubes and ovaries developing, the uterus, the vagina, and the broad ligament of the uterus. So all of those structures are visible here. And this differentiation basically is complete by the 7th week of development normally, okay? So that's what we are looking at here. So basically, what we have are these two ducts in that region, the mesonephric duct and the paramesonephric duct, okay? So the mesonephric duct basically goes on to form much of the male duct system, okay? While the paramesonephric ducts is what gives rise to the female reproductive system, anatomically that is, right? Okay, so what happens in a female? The paramesonephric duct goes on to develop while the mesonephric duct breaks down. So how I personally remember this is, if you look at mesonephric, like kind of sounds like starts with an M for men or masculine. So mesonephric duct gives rise to the male structures, okay? Paramesonephric ducts differentiate into female structures. So that hopefully would help you remember which is which. All right, so these are the changes, the formation of the genitalia that are happening along with the passage of time. So you can see here, weeks 8 to 20, quite critical as far as the formation of the reproductive system is concerned. So about week 7, in the presence of the SRY gene, which results in the secretion of something called the anti-Mullerian hormone. And this hormone is secreted by the sustentacular cells in the male. And what the anti-Mullerian hormone does is it inhibits the development of the paramesonephric duct, which would have turned it into a female structure, right? So instead of the paramesonephric ducts, the mesonephric ducts, again, they start with the letter M, so M for men or male. And so that takes place over the next 4 weeks or so, from 8 to 12 weeks, all right? And so these are the structures, the prostate, the bulbourethral glands, all of the accessory male sex glands form during that time. So then here we are looking at something called hermaphroditism. Hermaphrodite is an individual who has both male and female gonads, sex glands within the same individual. This is quite common in some of the lower organisms like earthworms, hermaphrodites. Some of the flatworms are hermaphrodites, right? So, but as you go to higher developments, higher levels of development, then the two sexes become quite distinct in two different individuals. Nonetheless, you have these cases where true gonadal intersex might develop. It's very rare, as you can see. It's about 1 in about 10 billion cases or something like that, right? So quite rare. So here you have an individual who has both ovarian and testicular structures, but usually none of them are functional. So the individual is effectively sterile, infertile, unable to reproduce, okay? And this can happen either in a genetic male, when I say genetic male, someone with an XY configuration, or a genetic female, okay? So that is true hermaphroditism, which is quite rare. Now, pseudo-hermaphroditism is where your genetic sex and your phenotypic sex do not match. So genetically, you might be female, but you might appear as male. So you're, for example, if you have an oversecretion of testosterone during development, even though genetically you are a female, an XX individual, your clitoris might grow to the point where it might be considered like kind of like almost a small penis, something called a micropenis. And so there might be some what is called ambiguous genitalia might arise. Or in males, if there is a lack of sufficient testosterone, and then the penis might not develop properly and it might be mistaken for a clitoris or something like that. So that would be an example of pseudohermaphroditism, as you can see here, all right? For example, 46XY. So genetically, you can see that this is a male, right? This is genetic male. However, the external genitalia resemble a female. Or you can have an 46XX intersex, where genetically this is a female, but the external genitalia are more along masculine lines. So this is what you're seeing here. So this could result, as it says, from things like congenital adrenal hyperplasia, right? Where your adrenal glands are hyperactive and hypersecreting male hormones, androgens, and that can give you this kind of a situation as well. All right, so then, so here you're looking at the development of the genitalia, right? Female on the left-hand side, male on the right-hand side. So it's very similar if you look at from the 6th to the 12th week, right? Very similar. So if there's fusion of the labia majora are homologous to the male scrotum, right? And then, of course, the clitoris and the penis, they're homologous structures as well. So you see how they're similar. By week 20, you can see the differences. So the labia majora and the labia minora are quite apparent here. Here's the clitoris. In males, the labia majora, what would have been labia majora in females, have joined together to form the scrotum, and then a penis has developed here. So these changes become clear by 20 weeks of development. Puberty, we already talked about that. That's when you become reproductively viable, right? And for the age of puberty is basically decreasing in both males and females, as we are seeing. And much of that, we talked about all of the different factors that are responsible for it, from diet to hormones to estrogen-containing substances in our environment. All of these things are giving rise to the fact that children are hitting puberty earlier and earlier. So that's what we're looking at here. So how it starts, so again, we mentioned it before as well. Your hypothalamus starts secreting GnRH, which is gonadotropin-releasing hormone, which acts on your anterior pituitary, making it release FSH, which is follicle-stimulating hormone, and LH for luteinizing hormone. And they are basically responsible for causing the pubertal development in both boys and girls. And these are the secondary sex characteristics. In females, growth of the pelvis, development of the breast, start of menstruation, all of those are signs of puberty, while in boys, deepening of the voice, growth of facial and body hair, growth of genitalia, muscular growth, all of those are seen here as secondary sexual characteristics, as you can see. So again, we talked about how African-American girls on an average are hitting puberty significantly earlier than other races. We see that too. Girls reach puberty about a couple of years earlier than boys, something else. So 8 to 12 years for girls, the age of puberty, and in between 9 to 14 years for boys. In some individuals, you'll see precocious puberty, which of course means puberty before the average, time. And this could happen because of things like, let's say, a tumor in your adrenal glands that is causing a hypersecretion um, uh, of androgens or sex hormones from that area, or a tumor in the testes or in the ovaries that can cause, or even a tumor in your pituitary gland, right, which is causing a hypersecretion of sex hormones. All right, so then menopause is, of course, the time at which the female ovarian reserve is exhausted. No more ovulation takes place there. She runs out of viable eggs at that time. So perimenopause is the time, the years surrounding menopause, right? So different women have different signs and symptoms, sometimes none. Things like irregular or skipped periods, they become more common in the perimenopausal years. And then menopause usually happens between 45 to 55 years of age. It's much determined by your genetics. So what your female first degree blood relatives, what ages they hit menopause is likely when you will hit menopause as well. So atrophy of your reproductive organs and breasts is what happens as well because of a withdrawal of the sex hormones, estrogen and progesterone, of course, vaginal dryness, and the a decrease in the thickness of the vaginal walls. All of those changes are seen as well, right? Some other signs and symptoms, hot flashes. Uh, some females will experience that with excessive sweating. Again, it's because of the sudden drop in hormones, especially progesterone, thinning of the scalp hair, but an increase in facial hair. Your risk for osteoporosis, having weaker bones, goes up because, again, estrogen is needed. The sex hormones are needed for healthy, sturdy bones. And so you might consider hormone replacement therapy at that time, but be aware of the side effects associated with it. So things like an increased risk for breast cancer, for uterine cancer, for ovarian cancer, right? Also for blood clots, for weight gain, for migraines. So all of those are potential side effects to initiating hormone replacement therapy. So that is something that should be discussed with your primary physician. All right, so then male climacteric or menopause or andropause, as it's called, basically is when the male secretion of testosterone plummets. And this is highly variable. Not every male goes through the same stages here, right? Usually starts in the 50s, around about age 55, when there's a reduction in testosterone. Actually, it starts much earlier. So age 30 onwards is when your testosterone levels start to go down about 1% each year. So by the time you are 50 years old, your hormone testosterone levels are already 20% less than what they were at 30. So this keeps going on. Most men do not have many visible or noticeable signs and symptoms. Rather, those who do might have things like lethargy, lack of energy and drive and motivation, loss of body hair, loss of muscle mass, decreased libido, sex drive, those kinds of things. Prostate problems become more common as well, right? Like problems with urination because of benign prostatic hyperplasia. We talked about that before as well. The risk of prostate cancer starts to increase with aging as well, right? Erectile dysfunction becomes more common, impotence with time. So this is simply an aging related effect, right? So not everyone should be prescribed testosterone. So this was all the rage a few years back, right? Testosterone replacement therapy, till we realized that it was probably doing more harm than good. So testosterone replacement therapy, or TRT, certainly is recommended for people who have a genuine reason for using that. For example, people who have primary hypogonadism or hypopituitarism, where the pituitary glands or their gonads are not secreting normal hormones as they should, right? So they would be legit candidates for TRT, testosterone replacement therapy. But someone who is just like 70 years old and is not getting an erection like he used to when he was 20, and that's very much expected, right? That's normal aging. So the person doesn't necessarily need testosterone. What they need is to keep up with a healthy lifestyle, healthy diet, exercise, right? All of those things that we have talked about before. Because testosterone replacement therapy has, many studies have shown that there's a high potential for heart disease, right? Coronary heart disease, as well as strokes. So people who were getting testosterone replacement therapy, men, were about anywhere from 5 to 8 times more likely to develop heart disease, stroke, right? Hypertension, blood clots, right? So a whole range of pretty serious side effects. So we have pretty much gone off of that trail. So the testosterone replacement is no more the cure-all, the panacea, as it was touted to be a few years back. All right, so what gene normally present on the Y chromosome is responsible for a male phenotypic sex? The SRY gene. Which duct system persists in female embryos? Paramesonephric duct. In male embryos, mesonephric, M for male, right? So what is the fate of the paramesonephric ducts in a female embryo? It forms the female reproductive structures like the uterus, the vagina, the fallopian tubes. What hormone do the tentacular cells secrete in the male embryo? The anti-Mullerian hormone, as we saw. And how does this affect internal genitalia development? It causes the mesonephric duct to develop rather than the paramesonephric duct. Okay, what causes the differentiation of tissues into male reproductive structures? Again, the anti-Mullerian hormone, um, the surge of testosterone, right? What factors affect the age that menarche first occurs? Your race, your diet, your body mass index, your genetics, your environment. All of them have to play into it. How do the female and male reproductive systems differ with respect to aging? Females have this definite time called menopause when they stop ovulating and menstruating, and they're reproductively not viable at that point, while males have a more gradual decline and they basically never lose their ability technically to produce viable sperm. So that's a major difference in between the two. All right, so we are done here with chapter 28. So on to our last chapter 29, which has to do with pregnancy and development of the human organism. So we are looking at the prenatal period here, of course, which begins with fertilization, the meeting of the sperm and the egg, of course, right? And it ends 38 weeks later with birth. So this is your gestational period of gestation or development, right? So now you have three shorter periods here: the pre-embryonic, the embryonic, and then the fetal stage, okay, or the fetal period. So here in this picture, you can appreciate exactly what we're talking about. So here you have fertilization taking place. You can see the sperm fertilizing the egg, then a zygote, which is a fertilized egg. Then it splits into a 2-cell stage, then a 4-cell stage, then a morula, which is a 16-cell stage, still a solid ball of tissue, but then it becomes a blastocyst. And at the end of about the second week, a blastocyst is this hollow ball of cells, and it's also differentiating into different tissue types. So differentiation has begun here, right? So then this is the stage of gastrulation from week 4 onwards. Gastrulation, where you're forming these different layers of cells. So this is a gastrula. By week 4, this is the embryonic period here, right? So you become a week 4 embryo, then week 6, then the fetal period starts, week 8 onwards, all the way till birth, okay? And then, of course, puberty, and then reproduction, and then the whole cycle starts all over again. So what distinguishes the pre-embryonic period from the embryonic period? In embryonic period, tissue differentiation or cell differentiation is taking place. In other words, the stem cells are turning into organs. That's what happens into the embryonic period. In pre-embryonic period, you just have a solid ball of cells without differentiation. So what are the changes from the embryonic to the fetal period? The embryonic period is when organs are still developing. In a fetus, which starts after 8 weeks of development, you have well-formed organs, and then those organs continue to develop till the time of birth, okay? All right, so then, all right. Let's try to load this up again. All right, there we are. Okay. So fertilization, of course, is when the sperm fertilizes or meets up with the egg. Each has 23 chromosomes from mom and dad, of course. When they come together, then you get a diploid zygote, which means 46 chromosomes here, right? The sex of the embryo is also determined here, because if the dad contributed an X chromosome for pair number 23, now this is going to be a girl, an XX configuration. If the dad contributed a Y chromosome for the 23rd pair, now this is going to be a boy. All right, so this happens. Where does fertilization take place? In the ampulla of the fallopian tube. We talked about that as well. The sperm are viable for 3 to 4 days inside of the female reproductive tract. If the sperm are outside of the body, just ejaculated, but not inside of the female reproductive tract, they die within 24 to 48 hours. But if inside of the body of the female, they're viable for much longer than that. So then something called capacitation takes place here, right? And capacitation, basically, as you can see here, are the changes, physiological changes in the sperm that make it capable to fertilize the egg or the secondary oocyte. Now remember, there's only one sperm that makes its way successfully and fertilizes the egg successfully, right? Out of upwards of 200 to 300 million sperm. So that sperm has to basically undergo these changes called capacitation so that it can fertilize the egg successfully, all right? Sometimes, rarely, what happens is that two sperm make their way into the same egg, right? So what's going to happen now? If you have like 23 chromosomes in the egg, and then you have 23 in one sperm, so that's 46, and then you have 23 more from another sperm, right? So what are you going to end up with? 46 plus 23, right? So that's going to be what? 69 chromosomes. So what you're looking at here, where two sperms simultaneously fertilize the same egg, or sometimes the egg is empty, no chromosomes because of this non-disjunction, and the sperm has the usual 23, so then you have only 23 even after fertilization. So what you're looking at here is basically something called molar pregnancy, okay? Molar pregnancy. And this is not a viable normal pregnancy. It's never going to turn into a normal child, obviously, because of the mismatch of the chromosome numbers. So molar pregnancies basically are stillbirths or abortions that you're looking at. A molar pregnancy is basically diagnosed on an ultrasound. So what you see on an ultrasound is what is called a snowstorm appearance. All right. So when you look at this picture, the ultrasonic picture, you don't see like anything remotely resembling a human embryo or fetus. What you see is like a snowstorm picture on the screen, okay? Highly suggestive. Another thing you'll see in molar pregnancy is highly increased levels of the pregnancy hormone HCG, right? Like 10 times the normal amount. That is highly suspicious for a molar pregnancy as well. And probably the most concerning of all, about 1% of all molar pregnancies actually turn into a cancer, something called choriocarcinoma. So it's quite a frightening thought that what should have been a happy, healthy, developing child, fetus, it actually turns out to be a massive cancer, tumor, choriocarcinoma, right? So all of those possibilities are there with that kind of a thing. Okay, so here we are after the sperm is fertilized, is introduced into the female reproductive tract. It swims towards the egg. All right, only one sperm is able to gain entry into the egg for fertilization. And for that to happen, it has to penetrate its way into the egg. And here you can see, so you see a number of sperm are surrounding the egg, only one gains entry. As soon as this one is able to penetrate the zona pellucida, this glycoprotein shell that the egg has, then some chemical changes take place that prevent the other sperm any further entry in there, right? So something called an acrosome reaction has to happen here. It's happening here, the acrosome reaction, as you can see, right? The acrosome reaction means this kind of like a helmet that the sperm had on its head, it kind of just dissolves away, and then the DNA from the sperm actually meets up with the DNA of the egg, right? So that's what the acrosome reaction is. So let's see. There, release of digestive enzymes from the acrosomes, the head of the sperm, and this allows the sperm to penetrate the zona pellucida of the egg, all right? And so that is what enables a single sperm to make its way into the egg. So what happens after that is cleavage. Cleavage is when the fertilized egg continues to grow and divide. All right, so here's the zygote, 2-cell stage, 4-cell stage, 8-cell, and then this is a morula, 16-cell stage, right? To this point, it was a solid ball of cells developing. Now it has become hollow, right? So this is something called a blastocyst. So this is the gastrulation or gastrula stage when it becomes hollow like this, right? So also in your early blastocyst stage, you see these two different masses of cells are differentiating. Differentiation has already started. So these blue ones are called the embryoblasts. They will turn into the embryo, the human embryo. And then the other cells here are called the trophoblast. They will turn into the placenta. Placenta is the tissue that is that supplies oxygen and nutrients to the developing embryo here, right? So the trophoblast is going to turn into the placenta. The embryoblast in blue here is going to turn into the actual embryo. So you're looking at all of those changes. So here, this is a good picture. This shows you the entire cycle, right? So the sperm swim all the way through the vagina, through the cervix, into the uterus. Then they swim all the way here into the ampulla of the fallopian tube. At the same time, the egg was released from the ovary. The fimbria sucked the egg in. The egg is traveling here. It got fertilized somewhere in the ampulla. Then the zygote moved for the next 7 days, approximately all the way down here, and then implanted itself into the endometrial wall, as you can see here. And on its way, you can see all of the changes, the fertilization taking place here, then the morula stage, then blastula, here's the blastocyst, and then it implants itself here, all right? Okay. So at this point, your trophoblast, which again was going to will eventually turn into the placenta, it divides into two layers. An inner layer called the cytotrophoblast and an outer layer called the syncytiotrophoblast, okay? So further differentiation going on. So at this point, the syncytiotrophoblast of the two layers, again, remember, this was the outer layer, that starts to produce HCG. HCG, of course, is your pregnancy hormone, right? Human chorionic gonadotropin. So these levels go up after the first week of a successful pregnancy, right? And what HCG does is it tells your body that a successful implantation and pregnancy has taken place. Therefore, it maintains your corpus luteum. Remember, what was corpus luteum? It was the empty follicle after it released the egg. So this empty follicle turned into the corpus luteum, and it's going to be maintained now because of the signal from the HCG being secreted by the syncytiotrophoblast, signaling a successful pregnancy. So the corpus luteum will continue to produce estrogen and progesterone. And what those two hormones will do is they will work on the uterus and keep it thick and vascular for a successful pregnancy to continue. And so these levels of HCG remain high for the first 3 months of pregnancy, okay? After that time, a placenta has successfully formed from the mother to the child. And so then now the placenta is secreting all the hormones, including estrogen and progesterone. So at that time, the corpus luteum degenerates and dies out, okay? So here you're looking at your blastula, which is this hollow ball. Again, this is the embryoblast, this is the trophoblast, and see how it is basically burrowing itself into the endometrial wall and literally sending out these finger-like projections to get its nutrition from the mom. These finger-like projections are called chorionic villi, right? So if hopefully you remember the villi, the finger-like projections in the small intestine. You have similar projections going on here from the implanted embryo into the mother. So chorionic villi are quite visible here, right? And at the same time, you can see the zygote developing further. Differentiation is taking place, right? So what do we have here? We have a mass of cells called the hypoblast, right? And then a mass of cells outside called the epiblast. Together, the hypoblast and the epiblast, they form what is called the bilaminar germinal disc, right? So cell differentiation is already started. You also have a yolk sac here. So the yolk sac, it's not like a bird egg. So the yolk sac in humans is where blood is going to form. So initial hematopoiesis or blood formation actually takes place here in the yolk sac because there are no bones yet, no bone marrow for blood to be made at this point, okay? So this graph shows you the fluctuations in your hormones with pregnancy. That's what you're seeing here. All right, so ovulation happened here, implantation around day 12, right? And so look what happened to your levels of HCG. They went sky high, right? HCG, you're pregnant. This is the pregnancy hormone, HCG. And then there is a gradual increase in progesterone, again, which is a pregnancy hormone, right? because the placenta is now producing both estrogen and progesterone. So this blue line is for progesterone, and then this pink one is for estrogen. So when estrogen and progesterone levels rise later in pregnancy after the third trimester, that's when your HCG levels go down. The HCG levels go down at that point. Okay, so here we are looking at some chromosomal abnormalities, and chromosomal abnormalities are surprisingly common. About 15% of all pregnancies result in stillbirth or abortion because of chromosomal abnormalities, 15%. So that's a significant number. The older the age of the parents at the time of conception, the higher the chances of having these chromosomal abnormalities. Okay. All right. So perhaps 50% of pregnancies that do get terminated are because of these, okay, because of chromosomal abnormalities. So too many chromosomes, but more commonly less chromosomes. Monosomies are more lethal than trisomies, as we mentioned before. So having lesser chromosomes than the normal number is a much greater risk factor for abortions and for stillbirths. All right. So then moving on, we talked about the hypoblast and the epiblast here. These are some of the extraembryonic membranes that are continuing to form, like the yolk sac. Now remember, the yolk sac, even though it's called that, but not like, unlike birds and reptiles, it does not store yolk. Instead, what it does, it forms blood cells, right? So this is a place where blood formation or hematopoiesis is taking place. Then you form another membrane, extraembryonic membrane called the amnion. And this would give rise to the amniotic cavity, which is filled up with amniotic fluid, in which the baby is suspended, and the amniotic fluid acts like as a cushioning agent to absorb, as a shock absorber, and also as a place where the baby is basically exchanging gases and nutrition from the mom, right? So when your water breaks, when they say that, it's actually the amniotic fluid. The amniotic cavity bursts, and the water flow is actually your amniotic fluid escaping at that time. And then the third extraembryonic membrane is the chorion. The chorion will eventually form the placenta. The placenta, again, is the tissue that connects the mother to the child. All right, and this is where food exchange and oxygen gas exchange takes place. So a placenta is a highly vascular structure. The placenta could be considered an endocrine gland, actually, right? So because it secretes so many different hormones, just like your other endocrine glands. So placenta is a quite an active endocrine structure that we are looking at here. It produces hormones like estrogen and progesterone, of course, which maintain the pregnancy and keep your uterus thick and fleshy and vascular to support and nourish the developing embryo. But in addition to that, the placenta secretes many other hormones as well that we would look at shortly here, like a thyrotropin, a hormone that causes your thyroid gland to function extra fast because now your metabolism has to really ramp up to keep up with the demands of the growing baby as well. So hypothyroidism is quite common after pregnancy, after you deliver a child in women because of the extra demands on your thyroid gland. So a good diet supplemented with iodine in your salt is quite recommended, quite necessary during pregnancy because, again, the pregnancy places a lot of demands on your thyroid gland as well, right? Okay, so these are what the some of the structures that make up a placenta, right? So the umbilical cord, as you can see, the umbilical cord carries two umbilical arteries which carry deoxygenated blood, even though they're called arteries, but they're carrying deoxygenated blood from the fetus to the mother for cleaning up and for oxygenation, and a single umbilical vein, which is carrying blood from the mother, oxygenated blood, even though it's a vein, oxygenated blood from the mother to the child, okay? So then we talked about chorionic villi, those finger-like projections that the embryo is projecting into the mother's tissue for gas and nutrient exchange, right? So what happens to the placenta is after birth, it is expelled from the body in the form of something called the afterbirth, okay? Because at that point, the baby has to feed on its own and it's breathing on its own as well, right? So, and also the placenta is a selectively permeable structure, which means it allows certain things to pass across, like respiratory gases, some microorganisms and hormones can pass across the placenta, others can't as well. So a couple of clinical conditions that we will talk about here. Let's talk about something called placenta previa, and then let's talk about something called abruptio placentae. Okay, so what are these different clinical conditions in pregnant women? Placenta previa is a placenta which is basically developed in such a way that it's blocking the exit from the uterus. It's either completely or partially covering the cervix, the os cervix region. So you see the problem here. So when the baby starts to exit the uterus at the time of birth, because the baby's head is going to rip the placenta from the wall of the uterus, right? Because again, the head has to go through the cervix, and so, but the placenta is covering the cervix, the placenta rips, and now the baby is in a real danger of dying because it's not quite born yet, and it has lost its lifeline to the mother. So that's placenta previa, right? It could be either complete placenta previa or partial placenta previa. If it's a partial case, then you can attempt a normal spontaneous vaginal birth or delivery. If it's a complete placenta previa, then usually you go for a cesarean section. All right, you have to go for a C-section because the child is not going to survive when it rips through the placenta. So that's placenta previa. Abruptio placentae, what is that? This is when the placenta detaches from the wall of the uterus before the birth of the baby. All right, so a lot of bleeding is possible here. Bleeding which can basically endanger the mom's life and certainly the life of the fetus as well, right? So abruptio placenta, what might cause the placenta to rip away from the uterus before birth? The chief cause is hypertension, high blood pressure in the mom. So if you have significant hypertension, especially if you have things like eclampsia, preeclampsia, right? So then you have to be careful about these kinds of things. Hypertension causing abruptio placentae. Something else, trauma to the belly region that can cause abruptio placentae as well. And whatever the cause, you have to deliver the child and stop the bleeding in the mom. All right, pretty much like placenta previa. So some people also advocate what is called placentophagy. If the rumors are to be believed, then even Kim Kardashian engaged in it. Placentophagy is like eating your own placenta, which is quite common in the animal kingdom. If you look at these savanna mammals like deer and elk, not elk, deer and antelopes, and certainly carnivores, they are known for eating up their own placenta after birth, which serves a number of purposes on the savanna, right? So in the savanna mammals, eating the placenta provides nutrition to the mother, who has been, a lot of demands had been placed on her body during the course of the pregnancy, number one. Number two, it cleans up the scent of the newborn, because a lot of predators are basically they hunt by means of the sense of scent, right? They smell newborn deer and antelopes and things like that. So by eating up the placenta, they're clearing up their tracks, right? So it cleans up the scent, and it also basically cleanses the area. It prevents infection and microorganism growth on the placenta. So nutrition, cleaning up the scent, recycling slash sanitization. That's why animals do it. Humans need none of that, right? We don't need to clean up our sands. We don't eat placenta for nutrition. There's no problem with recycling. And sanitization, humans usually do it. So no particular reason why anyone should be eating their own placenta. If anything, it can cause some bloodborne diseases to be passed through that, okay? So speaking of bloodborne infections that can pass through the placenta, remember the acronym torches, okay? So these are the microorganisms that can pass across the placenta and infect the child. So the TO in torches stands for something called toxoplasmosis. So Toxoplasma gondii, toxoplasmosis, is a parasite that is found in cat litter. So that's why they say why cats and pregnant women are not a good combination, right? So cat litter, the eggs of this parasite. And so what this parasite can do is it can pass across the placenta and form cysts in the brain of the developing embryo and in other tissues, leading to stillbirth and abortion, those kinds of things. The R here is for rubella, also called German measles. Rubella can also cause birth defects. So it's a teratogen, right? Teratogens are those substances that can cause birth defects in children, like nicotine, like alcohol, drugs of abuse, right? So rubella can do the same. Thankfully, we get the MMR vaccine, measles, mumps, rubella. That's what it stands for, right? Which protects us from that. The C is for cytomegalovirus. Again, this is a virus that has the ability to pass across the placenta and infect the unborn child. And then HE, they stand for both HIV and hepatitis B and C. So both of these viral infections can pass across the placenta. And finally, the S is for the sexually transmitted disease, syphilis. Syphilis can be passed on through the placenta too, and the child is born with different defects like what is called Hutchinson's teeth, malformed sharp teeth, which are sign of congenital syphilis infection. You might see that in the child. All right. So Torch's infection is what you have to remember. So that also reminds me of a rather graphic episode that I had when I was doing my resident, and this was overseas in India. So when I was doing my medical residency, my first year as an intern, and I happened to work in a rural hospital where they were seriously short on equipment. I was in the OB-GYN unit, and it was not uncommon for some of the orderlies there, if they had a surplus of placentas, human placentas, they would just kind of just toss them over the wall. So just like the savanna mammals, right? Not much sanitary thought there. And I've actually seen this with my own eyes, where there would be like stray cats, and they would literally come and they'll pick up and sometimes fight with each other over those placentas and take chunks and pieces out of it and devour it there, right? So it was, but then again, they cleaned up the whole thing. So it was a whole ecosystem, a whole food chain, a nutritional cycle that was in effect there. So my experience with placentas in OB-GYN rotation. All right, so moving on from there. So we talked about those. All right, so here we are looking at the developing embryo and the placenta, right? So these are the extraembryonic membranes. You see the amniotic cavity, right, which is filled up with the amniotic fluid. All here. There's the developing embryo along with the yolk sac. Those are the chorionic villi, finger-like projections for absorbing nutrients and gas from the mother's tissue. This is the chorion, which will eventually become the placenta, all around here. For the development, again, amniotic cavity, developing embryo, chorionic villi, yolk sac, chorion turning more and more into the placenta. And so here you have developed a fully, here's the placenta, right? So now the chorion has completely developed into the placenta, as you can see, and the embryo has developed as well. You see the yolk sac is diminishing at this point. Okay, so what are some factors or events that can prevent sperm from reaching the secondary oocyte? Well, a blockage in the male reproductive system, right? For example, or in the female reproductive system, like tubes that have been tied, tubes that have been scarred due to surgery or radiation, right? A hymen, which is impenetrable, right? Hymen is a membrane that guards the opening to the female reproductive tract found at the vulva, the opening of the vagina, right? So any or all of these factors can prevent a successful journey for the sperm to the egg. What are the three events that occur during fertilization? So the acrosome reaction, the capacitation, and then the actual fertilization. How many cells are present initially in the morula? 16. What are the two cell layers of the blastocyst? Two cell layers would be the syncytial trophoblast and the trophoblast, right? The trophoblast and the the cytotrophoblast and the syncytiotrophoblast. What would happen if a mutation prevented development of the syncytiotrophoblast cell layer? Then there will be no HCG, pregnancy hormone secretion. This pregnancy will be aborted at that point. Why is detection of significant levels of HCG a reliable test for pregnancy? Because that tells you that there is an embryo whose syncytiotrophoblast is secreting high levels of HCG. What are the two cell layers of the bilaminar germinal disc? A hypoblast and an epiblast. Which cell layers give rise to each of the three extraembryonic membranes, right? So the outer cell layers of the syncytiotrophoblast give rise to the three extraembryonic membranes, which would be the amnion, the chorion, and something called the allantois, ALLANTOIS. What are the main functions of the placenta? The exchange of nutrients and gases between the fetus and the mom. So gastrulation, this is, again, the stage at which the developing embryo is differentiating into different cell layers, right? It's becoming a hollow ball of differentiated cells, three-layered structures called an embryo. So how does that happen is what we can see here in this picture. You can see that these three layers of cells are forming within the embryo, one on the outer side, one on the very inner side, and one in between, all right? So here, now you can see they have been color coded for you in three different colors. The blue one is on the very outside, the most external layer of all. And this one is called your ectoderm. This one in the middle, the one that is colored red, is called the mesoderm. And then the innermost, which is in colored yellow here, is called the endoderm, okay? So ectoderm, mesoderm, and endoderm. So this is what gastrulation is all about, okay? Here you can see ectoderm, mesoderm, and the endoderm. And then the embryo kind of folds onto itself. So then now it has a head end and a tail end after folding, okay? Transverse folding here, it has folded up. So now you have these three layers, ectoderm, mesoderm, endoderm, arranged in a ball-like configuration, as you can see here. Okay, so next up, we are looking at what types of organs are formed from each of these three layers, right? So it's easy to remember, like if you think about ectoderm being the outermost layer in an embryo, it gives rise to all of your outermost organs. What are those? Skin, of course, it's the outermost, the largest organ. So skin comes out of the ectoderm. Also your brain and spinal cord, your central nervous system comes out of the ectoderm. Also your teeth, the enamel, the edges of the teeth and the lens of the eye, and your adrenal medulla, all of these structures are formed from the ectoderm. So how about the mesoderm? So in that list is all here. Then what about the mesoderm? So meso means middle. So the middle layer gives rise to the middle structures. What are the middle structures? Just as you go underneath the skin, fat, adipose, that comes from mesoderm. Blood, even that is a connective tissue that comes from mesoderm. Bones, muscles, cartilage, tendons, ligaments, connective tissue, all of that comes out of the mesoderm. And so then how about the innermost layer called the endoderm? The innermost organs, which would be your digestive organs, they come out of the endoderm. So in this picture, you can see what organs develop from each of those three layers. And this is called organogenesis, which literally means organ development, right? Okay, so then so we were talking about teratogens, and these are chemicals or factors that can cause deformities in the developing embryo, right? And the danger time here in terms of teratogens is week 4 to week 12, because that's when organogenesis is taking place, which means all of the major organs of the embryo are forming at that time, right? So you have to be especially careful. Things like cigarette smoking or nicotine exposure, alcohol, drugs of abuse, or even infections with the torches infections that we mentioned before. All right, so then let's see what events occur during gastrulation. The three primary germ layers form in the developing embryo. What are the locations of the three germ layers? The ectoderm on the very outside, the mesoderm in the middle, and the endoderm is the innermost. How are the results of cephalocaudal folding and transverse folding different? Cephalocaudal folding, head to tail folding. Transverse folding is when the three layers, basically the the three primary germ layers line up over each other. What structures form from each of the three germ layers? We talked about those: ectoderm, skin, CNS, so on. So why is it important? For a pregnant woman to consult with a physician before using any medication. Some of the medications could potentially be teratogenic, causing birth defects. So that is the reason why some caution is warranted here. All right, fetal period. So third month of pregnancy onwards till the time of birth, right? So rapid growth of the body takes place and the organs continue to mature during the fetal period, as you can see. So the average length is about 53 centimeters for the fetus at the time of birth, okay? And the weight increases the most noticeable during the first 2 months, as we see. So here we are looking at some of the causes for infertility and what can be done about it, right? So infertility can be caused because of a number of reasons. Endocrine causes like primary hypogonadism, primary ovarian failure, uh, or there could be secondary causes as well. For example, as you see here, blocked uterine tubes, either due to medications, surgery, even radiation exposure, right? In pelvic inflammatory disease, which is basically an STD gone bad. That's what it is, right? Ovulation disorders in males, anti-sperm antibodies, right? Or very low sperm counts that can happen as well. Abnormal sperm, something called azoospermia, okay? So any of those reasons. There are multiple treatments that are possible for each. So for example, if there is a blockage in the fallopian tubes or blockage in the movement of sperm from the male to the reproductive, male to the female reproductive tract, you can think of things like IUI, which stands for intrauterine insemination, placing the sperm directly into the uterus of the female, right? Oral medications like Clomid, these would promote ovulation, so thereby making pregnancy more possible. In vitro fertilization, or test tube babies as they're called, taking the sperm and the egg, putting them in a petri dish rather than a test tube, and allowing fertilization to take place there. Or donor eggs, getting eggs from a donor individual and then using those for fertilization. The fetal period is characterized by what changes? Organogenesis, growth, rapid growth, and further development of the different organs. So these are the three trimesters of pregnancy that we are looking at here, right? First 3 months is when the zygote turns into the embryo and then into the fetus. Second trimester, 4 to 6 weeks, 4 to 6 months rather, and of course the growth continues here. Third trimester, or the last trimester, is from the 7 to 9 months, and at that time, the mother's body is getting ready for the birth. So what are some of the hormonal changes that take place throughout this gestation period? The levels of estrogen and progesterone, right? They continue to rise. First, they're produced by corpus luteum during the first trimester, then the placenta takes over from that point onwards. So high levels of estrogen and progesterone suppress the levels of FSH and LH being secreted from your anterior pituitary. So that's kind of like a birth control. You cannot get pregnant again at that point. All right. Moreover, they cause bodily changes like growth of the mammary glands, the breasts, the uterus, all of those organs grow. The hair and the nails grow faster as well, right? The uterus is quite fleshy and vascular. Relaxin is produced by the placenta and the corpus luteum, which further causes blood vessel growth and vascularity in the uterus. So you can see the placenta, as I was mentioning, is very much an active endocrine gland. It's secreting a number of hormones like this one, CRH, that stands for corticotropin releasing hormone, right? And what it does is it causes secretion of aldosterone from your adrenal glands. Aldosterone causes salt and water to be retained, and therefore the edema, the swelling that you notice during pregnancy. Then HCT, human chorionic thyrotropin, this stimulates the pregnant woman's thyroid gland to secrete more hormone for the baby as well. And that is the reason why hypothyroidism is so much more common in females after birth, because birth itself, pregnancy places a lot of demand on the thyroid gland, right? So she might be a good candidate for thyroid supplementation with drugs like levothyroxine as an example, right? Or Synthroid. Now, a lot of times, postpartum depression, right? It's common depression in women after giving birth. But this could very well be not a psychological, but an endocrine issue. Postpartum depression could be due to hypothyroidism. So always check for thyroid levels in a postpartum female, someone who has just given birth, all right? So if the hormone levels from the thyroid gland are low, then you can literally fix that depression by prescribing Synthroid to the patient, right? Supplementing the thyroid hormones, and she will feel much better after that. Also, human placental lactogen, which of course has the name lactate from milk. This promotes lactation, as you can see here, okay? Prolactin, again, having to do with lactation, right? It promotes milk production. Oxytocin promotes milk expulsion, what is called the let down reflex. So that happens as well. The uterus continues to expand, which might cause urgency of urinary urgency, because the growing placenta, and the uterus are placing some pressure on the urinary bladder. It can also cause constipation because it's pressing backwards onto the rectum as well, right? All right, so all of those changes, as you can see. So the mother's literally, her organs are being cramped for space because the fetus is growing so remarkably at this point, all right? So the fundus of the uterus reaches about the level of the belly button, the umbilicus, by the 22nd week at this point, all right? So these are changes taking place in the breast during pregnancy. Under the influence of the melanocyte stimulating hormone, which again comes from the placenta, the areola, the skin surrounding the nipple, becomes darker. And also a dark line called the linea nigra, it develops in the pelvic region of the female as well, all right? So here in this picture, you can see 4 months, 7, and then 9 months. There's a fully developed fetus ready for birth, and all of the changes that are taking place here. So other signs and symptoms like morning sickness, they appear as well, and they have to do with the fluctuating levels of hormones, like levels of steroids, like estrogen and progesterone, the sex hormones, and some of them can even lead to things like gestational diabetes, like human lactogen, placental lactogen, that causes the mother's glucose levels to go higher to feed the child. But in susceptible women, this can lead to gestational diabetes. So if you have developed gestational diabetes during pregnancy, chances are you're going to develop full-blown diabetes type 2 even outside of pregnancy. So now would be a good time to go on a diet and keep a close eye on your blood sugar levels. So morning sickness happens in women, more so in some women during the first trimester, all right? In some, it would just be a little nausea. In others, a full-blown hospitalization would be required because so much vomiting will take place that they will end up with metabolic alkalosis in some cases. All right. Okay. So constipation, heartburn, indigestion, even hemorrhoids because of increased intra-abdominal pressure. All of these things are quite common at this time, right? And so the weight gain that happens during pregnancy is because of the, of course, the added weight of the growing fetus, also the breast and uterine and placental enlargement, edema, fluid retention because of high aldosterone levels, and extra fat that develops to protect and to nourish the child as well. So keeping in view all of these increased demands, you need about 300 extra calories per day, and also minerals and vitamins. So prenatal vitamins are needed to supply for folic acid. That one is important because a deficiency of folic acid can lead to developmental defects in the growing fetus, like spina bifida, right? The spine does not form properly. So make sure that prenatal vitamins are being taken here. Gestational diabetes, again, develops during the first pregnancy. And what happens again is high glucose levels and insulin resistance. Again, it's more common in women who have a family history of gestational diabetes, who are heavier, have a higher body mass index, or physically inactive as well. The same women who are at risk for preeclampsia also are at risk for gestational diabetes and also hypertension, high blood pressure. All right. And so there's always an increased risk of birth complications, C-section needed, again, for women with gestational diabetes, because these diabetic women tend to have children with big children, right? Children with the heads are quite large as well. So birth in complications are quite common in diabetic women. So C-section is by default more common here as well. All right, hyperemesis gravidarum, that's a term for the vomiting of pregnancy. In some cases, it could be severe enough to warrant hospitalization. And then you have to manage the body fluids and the pH of the blood as well. As I mentioned, metabolic alkalosis is what you will see here. What changes in your cardiovascular system? It becomes more efficient, right? So let's reload it all the way here. Right, so the cardiovascular system, talked about these changes needed. All right, so for the respiratory system, it may cause some dyspnea, difficulty breathing, again, because of the pressure in the abdomen due to the expanding uterus. Nosebleeds become more common in some women, okay? What else? The brainstem becomes more sensitive to carbon dioxide, so breathing becomes more efficient, and that facilitates gas exchange across the placenta, right? Increased tidal volume, pulmonary ventilation, oxygen consumption, all of those are increased. Urinary system becomes more effective as well, right? So your glomerular filtration rate, or GFR, increases from 30 to 50%. And urinary tract infections become more common at this time because, again, the growing, expanding uterus puts pressure on the ureters. So some general differences in the mother and developing embryo fetus between the first trimester and the second trimester: continued growth and weight gain, both in terms of adipose and weight, fluid retention. How do estrogen and progesterone act to sustain pregnancy? They make the uterus more vascular, increase blood supply across the placenta, breast development. All of those changes take place here. What are the actions of CRH, corticotropin releasing hormone? Increased aldosterone secretion, therefore fluid retention. Human placental lactogen, or HPL, more glucose in the mother's bloodstream to feed the child. Oxytocin for milk expulsion and also for initiating labor. And prolactin, again, milk synthesis. What impact does the growth of the uterus during pregnancy have on other organs of the abdomen? Heartburn, urinary urgency, more common UTIs, hemorrhoids, constipation, all of those things have become more common. All right. So then labor is when the baby is being born, of course. Typically, it's at 38 weeks for full-term pregnancy, okay? And if it takes place before 38 weeks, then it's considered premature labor. And then that is attended by a whole host of complications there. So not all contractions are true labor contractions. So you have false labor contractions as well, something called Braxton-Hicks contractions, as you can see here, okay? And how you know they're not the real deal is because they're relatively weak. They do not increase in intensity progressively. They're limited to the lower abdomen and the pelvic region, and no changes in your cervix. The cervix doesn't dilate or anything. So for true labor to happen, how that happens is, again, hormones do it. So the mother's secretion of oxytocin progressively increases from the posterior pituitary, right? So high levels of oxytocin, which act on the uterus, causing the uterus to contract more and more forcefully. And that, at the same time, your cervix softens and dilates more. So now that is what true labor looks like, basically. So these are some of the indications, 5 signs of true labor: increase in frequency of contractions over time, increase in intensity as well, pain, constant pain radiating, getting worse, and cervical dilation. The cervix dilates, okay? All right, so, and this is a positive feedback loop, okay? So what that means is, what is positive feedback? Well, as the baby's head presses and stretches the cervix, it sends the stretch receptors perceive it, send this response to the brain, and the brain responds by secreting oxytocin from the posterior pituitary and the hypothalamus higher up there. The oxytocin acts on the uterine wall, causing more powerful contractions. The more the cervix expands, the more stretch receptors communicate to the brain, the more oxytocin is secreted. So this is a positive loop that is developed, and it culminates in a successful birth, basically. So this is an example of a positive feedback loop. If you're 2 weeks past due date, right, after 38 weeks, then you might have to induce labor, and then you will give Pitocin, which is synthetic oxytocin, to initiate labor, as you can see here. These are some anesthetics that you might consider for labor pains. So what is most commonly used is an epidural nerve block, right? It's called that because the needle deposits the anesthetic here in the epidural space, and it numbs your genital region, right? The birth canal. It does not interfere with the contractions themselves. You have some other options like a pudendal nerve block, which is the minimal amount of anesthesia. It numbs only the lower vagina and the perineum, and you can still feel contractions in this. Spinal nerve block is reserved for C-sections, cesarean sections. So even the anterior abdominal wall is numbed here because you have to make an incision to get the baby out there, right? Okay, the 3 stages of true labor. First one is the dilation stage, when the cervix dilates, and it ends when the cervix is dilated to about 10 centimeters, all right? And it takes longer in women who have never given birth before, nulliparous women, 8 to 24 hours. In women who have given birth before, like parous women, it's shorter, from 4 to 12 hours, okay? Then is the dilation. Well, it's still the dilation stage. That's also when your water breaks. So this is amniotic fluid, because the amniotic sac ruptures, and so the water escapes at that point, okay? So early dilation, and then the late dilation stage, the positive feedback loop has been established here. Then comes the expulsion stage, which can extend anywhere from half an hour to several hours, depending, again, if you're a first-time birth giver or a multi-gravid woman. So, and pushing down, bearing down helps during this time. And crowning is when you can see the crown of the head of the baby emerging from the birth canal. Sometimes, especially if the baby has a very big head and the mother has a delicate, smaller pelvis, more common in diabetic women again, then you might have to perform an episiotomy, a surgical incision to widen the opening so the baby can be born more easily. Umbilical cord is clamped and tied off at that point. So here you can see the crowning taking place, right? So this is the expulsion stage. Then the placental stage, which can take place up till half an hour after birth. So this is when the placenta is also expelled, all right? And you have to be careful at this point to make sure that the entire contents of the uterus are removed, because if not, then you might be faced with RPOC, which stands for retained products of conception. And this can lead to ongoing hemorrhage or bleeding, right, of conception, which is quite a common cause of maternal mortality and morbidity in much of the developing world, right? Postpartum hemorrhage. Another special type of postpartum hemorrhage, especially in women who are anemic to begin with, is something called Sheehan syndrome. And what happens in Sheehan syndrome is that so much blood is lost at the time of childbirth, loss of blood at childbirth, where not enough blood is left to supply your anterior pituitary. So loss of blood at childbirth, leading to pituitary necrosis. Your pituitary gland breaks down because of lack of blood supply, leading to pituitary necrosis. So for this woman, again, you have to supplement all of the hormones, otherwise she won't ever be able to get pregnant or have a normal reproductive function, or any of the other pituitary hormones for that matter. So hormone supplementation is absolutely needed in Sheehan syndrome, right? So retained products of conception, Make sure you scrape them out. If they don't come out, then sometimes you might have to do a DNC, that stands for dilatation and curettage, where you literally scrape the walls of the uterus to make sure everything has been removed, okay? Some other techniques that can be used to facilitate childbirth in addition to C-section is what is called a forceps delivery, right? So you literally use a pair of forceps or tongs to basically pull the baby's head out, or you can do a vacuum suction, right? A suction device to facilitate childbirth. So some other options which are available. All right, so here is the placenta. You can see it has been clamped and cut and removed, okay? So sometimes the baby comes out bottom first, which is called a breech delivery, which has its own set of complications attached. Because of course, in breech delivery, the bottom end comes out first, the umbilical cord is detached, but the baby's head is still inside of the birth canal. So the baby can't breathe. So you have to make sure that the baby comes out in good amount of time. So again, forceps delivery or vacuum suction devices or whatever is needed to get the baby out in good time. So how do progesterone, estrogen, and oxytocin interact to eventually bring about labor? So we talked about progesterone as the pregnancy hormone, keeps the uterus nice and healthy and fleshy. Same with estrogen, also acts on the female breast for milk production, and oxytocin promotes the labor contractions. 5 signs of false labor: pain is not increasing, pain goes away with movement, it does not radiate, right? There's no cervical dilatation. 5 signs of true labor: pain is consistent, it increases in intensity, cervical dilatation, and pain is radiating. How does the positive feedback mechanism of true labor lead to birth of the fetus? We talked about that too, oxytocin, uterus. What are the 3 stages of labor? Dilatation, dilation stage, expulsion stage, and then afterbirth, or the placental stage. Why is the placental stage a critical part of labor? Because if the placenta is not completely removed, you can have ongoing persistent postpartum hemorrhage and infection of the uterus, which could be life-threatening. All right, so what happens in the child as soon as he or she is born? The first breath is taken within 10 seconds, right? The lungs inflate, and then they will keep working from that point onwards. And that could be a problem if you have a preemie. The child is immature, the lungs haven't developed enough, there is not enough surfactant, so the lungs stay collapsed. So in that case, you will need to assist the child, help it breathe. You have to introduce synthetic surfactant into the lungs to open up the lungs. Sometimes they need to be on a ventilator as well, right? So what changes happen here? Right after the newborn takes their first breath, the foramen ovale in the heart closes, so there's no intermixing of blood, oxygenated blood with the deoxygenated blood on the 2 sides, okay? All right. So when the neonate takes its first breath, the lungs expand, the foramen ovale and the ductus arteriosus close. How does the fetal circulation change once the neonate begins breathing? This is how it changes. The pulmonary resistance decreases, pulmonary arteries open up, decreased pressure on the right side of the heart, greater on the left side of the heart. So in other words, the 2 sides of the heart are separated now, no intermixing of blood. So what happens to the woman after she gives birth? Postpartum changes, estrogen, the hormone levels drop suddenly and rapidly, and that can lead to what is called baby blues, this depressed mood, right? And also hair loss is common because, again, that estrogen and progesterone support is gone. Corticotropin releasing hormone levels drop suddenly, okay? And that can also lead to depression. And weight loss happens as well. Urination increases as well, right? Fluid reduction, as you can see here. Amniotic fluid is expelled, then something called lochia, which is some blood along with mucus and endometrial tissue, which is expelled again through the birth canal. Can happen for several weeks following birth. Also, urination increases because the aldosterone is not there anymore, because the corticotropin releasing hormone is withdrawn at that point. So urination increases, lochia flow. So all of that leads to fluid reduction, okay? Also, profuse sweating happens as well to get rid of the extra fluid. So then lactation starts. So what comes out first is something called colostrum. Colostrum is not milk yet, but it's rich in antibodies like IgA antibodies specifically. So that's wonderful for the baby, so they can get passive immunity if you breastfeed them right after birth. So that's what you're seeing here, right? Okay. Breastfeeding is also protective against the mother. So breastfeeding is protective uh, against breast uterine uh, and ovarian cancers, right? Because it balances out the hormones, right? Uh, so there's also weight loss is much faster with it. So it has significant benefits, health benefits for the mother as well. Okay, milk let down reflex. We talked about that as well before. Oxytocin basically causes milk to be released. All right, so that's what we are looking at here. When you breastfeed, the child lactation also prevents high levels of prolactin, prevent ovulation, and they prevent uh, gonadotropin releasing hormone and FSH and LH release. And so therefore no further ovulation takes place. But it's not an absolute method of birth control. So even if you're breastfeeding, sometimes breakthrough pregnancies can happen. So you have to look at other methods of birth control there as well. After pains are contractions that happen, continue to happen for many days after you have given birth, okay? And because the oxytocin levels are still high and that continues to stimulate the uterus. Pretty much what happens during your menstrual cycle, the menstrual cramps. So which hormone levels drop in a postpartum woman? Estrogen, progesterone, all of the placental hormones. What happens as a result of this reduction? Mood changes, body weight loss, fluid loss, urination, sweating, all of those. What are 4 ways by which excess fluid is eliminated? By means of lochia, by means of profuse sweating, by means of increased urination, and by means of amniotic fluid escape. How does the positive feedback mechanism ensure that the infant receives breast milk? The more you put the breast to, the infant to the breast, the more the suckling action, the more oxytocin is secreted. So this is a positive feedback that needs to keep going for ongoing lactation. How are prolactin and oxytocin related to both the uterine changes and lactation? Prolactin is for milk synthesis, okay? Oxytocin is for the let down reflex for expressing expulsion of the milk from the breast. Oxytocin also causes birth, labor contractions. All right, so next up, we are looking at genetics here. And so some terms to look at in terms of human genetics: heredity, the science of inheriting characteristics from your ancestors. Genetics is the field of biology that studies heredity. Karyotype is an actual picture of your chromosomes, the 46 pairs of chromosomes. Homologous chromosomes are the first 22 pairs of chromosomes, which are very similar in size and shape. And they're also called autosomes, okay? So here is a human karyotype. You can see they're in pairs. There's 23 pairs total, and 46 chromosomes, that is, and the first 22 pairs are called autosomes because look how similar they are in size and shape. One from mom, one member of the pair from the dad. And if you look at the last pair, 23rd pair, you can tell me the biological sex of this individual, right? A much longer X chromosome and a much shorter Y chromosome. XY, that's a boy that you're looking at. So this is what a karyotype basically looks like. So sex chromosomes are the last pair here. XY would be a boy, XX would be a girl, as you can see. Alleles are 2 sets of genes, sometimes more than 2, that control the same trait. Like, for example, alleles for eye color. So let's say mom has brown eyes, so allele here for brown, and dad has green eyes, so a green-eyed allele on the other chromosome. So alleles are a pair of chromosomes or more chromosomes, pair of genes or a group of genes that control the same trait. That's what they are, all right? So dominant versus recessive allele. So before we look at these terms, let's go ahead and do some genetic crosses here. So that would basically explain what these terms mean, okay? So what I'm gonna do is, I'm gonna stop share for a minute so you guys can see my notebook clearly here, okay? So let's say you have a brown-eyed individual. Okay, so I'm gonna write down the brown-eyed genotype, and this person is crossed with another individual who is blue-eyed. All right, so you can see that the capital B here is for brown eyes, right? And the lowercase b here, all right, is for blue eyes, right? So a few questions. So why brown with capital B and why blue eyes with lowercase b? It's because brown eyes are dominant over blue eyes in males. In other words, if you have two alleles, two genes, one for brown eyes, capital B, one for blue eyes, lowercase b, the person will invariably will have brown eyes because brown is dominant over blue eyes, and therefore it overcomes and expresses itself. The only way that you can have blue eyes is you have two recessive alleles, right? So dominant versus recessive. Dominant expresses itself. Dominant genes expresses itself, right? While the recessive alleles are hidden. Okay, they're hidden. They are not expressed. So now you might be wondering, why did I write two letters, two capital B's for brown eyes or blue eyes? Why couldn't I just written a single B here, a capital B and a lowercase b here? The answer to that question is, this is a genotype, right? Genotype means you got one allele from each parent. We have two parents, right? Mom and dad. So one comes from mom, one comes from dad. Same for that. So therefore, you always write a genotype in two letters, one from mom and one from dad. So that is called genotype, right? Genotype is the uh, letter combination, right? Your letter combination or your genetic makeup. That's what genotype is. Okay, genotype are it's the letter combination or the genetic makeup. So then you have something else called a phenotype. Okay, a phenotype. So what is a phenotype? Well, if you look at the word itself, it starts with ph, phenotype, and you can think of that ph for physical as well. So a phenotype is the actual physical characteristics of an individual, right? For example, this person is brown-eyed. So brown eyes is a phenotype. The genotype is big B, capital B, capital. That's the genotype. Phenotype is brown eyes. For this person, the genotype is lowercase b, lowercase b. The phenotype is blue eyes, all right? So now, how do you make a cross between these two individuals anyway, right? How do you cross these two? So for that, we have to play a game of tic-tac-toe, and most people almost everyone has played that before. Only in genetic terms, a tic-tac-toe cross is called a Punnett square, okay? So we're gonna make a Punnett square, and we are going like this, right? There's a tic-tac-toe, Punnett square, and we are going to cross these two individuals together. So let's start with this person first, okay? So, so I'm gonna take each letter, like one capital B, I'm going to put that capital B in a box, this box right here. So one B here, right? And then the other capital B in the other box right next to it. I'm going to leave this one empty. I'm just going to leave this one empty. And then I'm going to go back to the blue-eyed parent, this one. So I'm going to write the lowercase b here, right? And on this side, and another b, lowercase b down there, right? So this would be here vertically, and one parent would be at the top horizontally. And then I'm going to simply go ahead and I'm going to cross these two here, right? So I'm going to write like capital B and then lowercase b next to it like this. And same here, capital B, lowercase b, right? And then same these two here. And then these two go together, right? So again, capital B, lowercase b. And then again, and for the last one, those two go together. So capital B and lowercase b, okay? So these are the four offspring that came out of this cross. Those were the parents on the two sides. These are the offspring. So the genotype for all of them turns out to be this, capital B, lowercase b. This is the genotype, right? So then what is the phenotype of these individuals? Capital B, lowercase b, right? The genotype is that. Remember, capital B is dominant. So brown eyes are dominant over blue eyes. These are all brown-eyed children that we have here, right? However, there's a difference in between this type of brown-eyed individual and these types of brown-eyed individuals, right? Here you have both capitals, right? Both are dominant. So uh, both of them, since they're the same letter, capital, it's called homozygous. They are homozygous brown-eyed people, right? Because again, homo, that means same. So both capitals. So where is it? This one is a homozygous brown-eyed individual because both are capital B's. Look at this one capital B, one lowercase b. So there's a difference, one capital, one lowercase. So that is called a heterozygous brown-eyed individual. Hetero means different, right? So heterozygous brown-eyed individual right here, right? This one is heterozygous brown-eyed, one capital B, one lowercase, two different heterozygous. This one here, that the parent, this one was a homozygous, same, capital B, homozygous brown-eyed individual. So based on this, now we know what genotype is, what a phenotype is, right? What is homozygous, what is heterozygous, what is dominant, what is recessive, how do you make a Punnett square, what is genotype, what is phenotype. All right, so that was for a basic cross here, right? So this was a basic Mendelian cross. However, there are some variations to it as well. Okay, so let's take a look at a variation which is called, which is a variation which is called incomplete dominance. So let's see what that is all about. Incomplete dominance. Okay. So here is an example. Let's take a dark-skinned individual, and we'll call that who's homozygous. So for let's say capital D is for dark skin. Okay. And lowercase d is for light skin. So we are here, as you can see, crossing a homozygous dark-skinned individual with a homozygous light-skinned individual. All right, here's the cross. And so how are we going to do it? We're going to make a Punnett square again. Here it is. Okay, there, tic-tac-toe, Punnett square. What I'm going to write, the dark-skinned individual here first. So capital D, right? And then the light-skinned individual vertically this way. So lowercase d. So in the first box, you're going to cross those two. Remember, capital D is dominant, dark skin is dominant. So it comes first. And then the second box, these two, right? The third box, those two again, capital D, lowercase. And then the last box, same, capital D, lowercase d. All right, so the genotype, the 100% genotype that you got for all of the offspring here is what? Capital D, lowercase d, so heterozygous. So you would be inclined to say that all of them are dark skinned, right? Because that's what we talked about, dominant and recessive. However, when you look at traits that display incomplete dominance, there is actually a mixture of the two. Or in other words, an intermediate phenotype comes out. So for this person would be brown skinned, all right? Intermediate shade, brown skin. So that is incomplete dominance. as we see here, right? So for some traits, you will not have dominant and recessive, rather you will have a mixing or an intermediate type of both. So this is what we are seeing here. So then you have another example of these genetic traits, something called codominance, okay? And the best example of codominance is seen in human blood types. So we're looking at codominance, which means two traits which are equally dominant or codominant, okay? So let's make a cross about among two individuals, one of whom is blood type AB, and let's say the other person is blood type O, okay? So here are the blood types. And remember, genotype is always two letters, one from mom, one from dad. So AB, and then OO for blood type O individual, right? Let's make this cross. So I'm going to write this parent at the top, right? So A here and then B next to it. This is the OO parent. I'm going to write the O here and then O here. So what do we get in the first box? We cross these two, A and O. The other cross, B and O. Then these two here, A and O. And those two again, B and O, all right? Okay, so what are the blood types that we see here? So as you can see that A was written before O. In this case, both A and B blood types are dominant over O blood type. So the first one here, the genotype is AO, but the phenotype, what it actually is still blood type A. So 50% of the offspring here are blood type A, are blood type A. They're heterozygous, but they're still blood type A. And then the other 50% are blood type B. They're heterozygous B. The genotype is BO, but phenotypically they are blood type B, okay? So let's make one more cross. Let's make a cross between these two individuals, AO and BO. So let's make this Punnett square here, right? So we are crossing these two individuals. So I'm going to write for this person, this person here, A here, and the O in the box next to it. Then for the other parent, B here, and O in the box at the bottom. So let's cross these two first. What do we get? A, B. Then what do we get here? B, O. For the third box, A, O here. And then for the last one, both are O's, O and O. Okay, so let's figure out the blood types of each of these here. This person is AB. So what does that tell you? Both A and B are equally codominant. None of A is not dominant over B, B is not dominant over A. They are both codominant. That's why it's called codominance. So this blood type would be phenotypically expressed as AB. This blood type again is A. The genotype is AO, but the phenotype is still A because A is dominant over O. This blood type is genotypically it's BO, but the phenotype is still B because blood type B is dominant over O. So blood type B. The last one is blood type O. So what you can appreciate in this cross is if you cross someone with blood type genotype AO with blood type genotype BO, you have a 25% chance of getting a child with all of the four known human blood types. AB, 25% chance of AB child, 25% of A child, 25% of B child, and 25% of O child, right? So this is the most genetically fruitful combination, AO and BO, right? So you have a 25% chance of all of the human blood types showing up here. So this is an example of codominance. Incomplete dominance was when you got an intermediate phenotype like dark skin and light skin, and you got brown skin as an example. All right. Codominance, two traits like A and B are both equally dominant. Okay, so let's see if we have any other type of crosses to discuss here. All right, so you hopefully know what heterozygous, homozygous, genotype, phenotype, dominant, recessive, incomplete dominance, codominance. We talked about that as well. All right, so yes, one final type of cross that we will talk about is something called sex-linked or X-linked inheritance, okay? And that would be the last example of genetic crosses that we will discuss. Okay, so so how do you write? Well, let me stop sharing here. So how do you write the genotype of a typical male? All right, so by now you know that, we discussed it. The genotype of a typical male is XY. And what is the genotype of a typical female? X and X, right? Simple as that. So let's make a very basic garden variety cross between an average male and an average female. So I'm going to write this guy, the dad over here. So one X goes in this box, Y goes in the box next to him. And then here's the mom, so X here, and then the other X down there, right? Let's cross them, those two, X and X. This and that would be an X and Y. Then these in the third box, X and X, and then this one in the last box, X and Y. So what you can see straight up here is in any cross between a male and a female, there's a 50% chance of the children turning out to be females and an equal chance, 50% chance of boys coming out, right? So there is no gender or sex discrimination here. There's an equal 50% chance of both, right? So now you have something called sex-linked traits or X-linked traits like red-green color blindness, which is more common in males, right? So in these people, they cannot differentiate between color red and color green, right? So it kind of looks gray all across. Or hemophilia. Now, this is a blood clotting disorder. If you have it, then you can literally bleed to death from like little cuts, okay? Or muscular dystrophies. So these are some examples of traits which are sex-linked or X-linked. Why are they called that? Because these traits, red-green color blindness and hemophilia and muscular dystrophy, are carried on the X chromosome, not on the autosomes, not on the 22 chromosomes, but the 23rd pair, the XY or the XX pair. That's where they are carried on. And remember, the Y chromosome, it does not carry any genes or alleles. It's only there to make you male. All right. So all the other traits are carried on the X chromosomes only, exclusively. And that's why they're also called X-linked traits, sex-linked or X-linked traits. Why sex-linked? Because they are found on the sex chromosomes. And why X-linked? Because they're only found on the X chromosome. The Y does not have any trait. So let's talk about color blindness, right? And we're going to denote color blindness with the small letter C, all right? So little c is for colorblind, like that. So let's make a cross between a colorblind male and a normal female, okay? So how do I write a colorblind male? First of all, let's write a male, so that would be X and Y. So what I'm gonna do is to denote that this male is colorblind, I'm gonna write a small c on top of the X. Only X, remember, Y does not have any genes, any alleles. It only makes you male, all right? And the mom is normal. So two normal X's for mom here, right? So let's make this cross here. So, all right, so I'm gonna write the dad at the top, X, C, Y, and I'm gonna write the mom on the side. So here's the Punnett square again for you, right? Dad and mom. So let's cross the first two. Turns out to be what? X, X, little c, correct? Then these two, X and Y, right? And then this and this, again, X, X, C. And then the last one is X, Y. Okay, what did we get out of this cross between a colorblind dad, colorblind male, and a normal female? Look at all the females, the daughters born to this couple, they're XXC. What is that? Are they colorblind? Are they normal? Or are they something called a carrier where they themselves do not have color blindness, they have perfect vision, but they can pass on that trait to their children? Certainly, that's what it is. So these are carrier females, all right? They're carriers, which means, again, they do not have the disease themselves, but they can pass it on to their male offspring, okay? XXC. So all of the daughters born to this couple would be carriers. How about all of the boys born? Normal males, right? Okay, so now let's go ahead and let's cross these two individuals, a carrier female with a normal male. Let's see what happens here. So if you have a normal male, so that would be XY, right? And then crossed with a carrier female with a little X there. Normal male and a carrier female. Let's make the Punnett square here. And I'm going to plug in all the letters as before, right? Dad, mom. So let's take a look at the first box, X and X. Second box is X and Y, right? Now in the third box, XXC, right? And then in the final box, XCY. Okay, so let's look at the daughters first. What do we see with the daughters here? These two are the daughters, again, XX, right? So 50%, half of the daughters would be completely normal, XX. 50% of the daughters would be carriers, XXC, which means they have normal vision themselves, but they do have the trait for color blindness that they can pass on to their children, right? So 50% of the daughters are normal. 50% of the daughters, half of them would be carriers. How about the boys? 50% of the boys are perfectly normal, XY. The other half are colorblind. Remember, in males, all it takes is just one messed up X chromosome, 1C here, and the Y doesn't do anything. So the 50% of the boys are still colorblind here. But for the females, these are carriers. They're not colorblind themselves, but they can pass it on. All right. So there you go. This is what happens here. So let's do one more cross. Let's cross these two individuals together. So a carrier female with a colorblind male. Okay. So colorblind male is XCY. So what lesson did you get here? Males cannot be carriers. They either are perfectly normal or they do have the disease, right? They cannot be carriers. Females have three choices. They can either be normal or carriers or colorblind, okay? That also explains why more males are colorblind or have hemophilia or have muscular dystrophy as compared to females, because all they need is one messed up X chromosome and they have it. Females need two of the messed up X chromosomes to get the disease. All right, so we were crossing a colorblind male with a carrier female, which is this colorblind male with a carrier female. Let's make this cross. So I'm going to make the Punnett square, right? And I'm going to write the dad here, X, C, and Y, and the carrier mom on the side. So here's the colorblind dad at the top and the carrier mom on the side. Let's make the first box here, XXC. Second box is X and Y. Third box is XCXC. And the fourth box is XCY. Okay, so what did we get here? Let's take a look. So among the daughters, 50% are carriers, XXC carriers. How about these? The other half are colorblind daughters. So there, when a colorblind male marries a carrier female, half of the daughters are carriers. They're normal, but they do carry the allele, and half of the daughters will be colorblind. How about the boys? Again, half of the boys are completely normal, half of the boys are colorblind. So there's a 50% chance of either colorblind child, whether daughter or son, in this genetic combination. All right, so this is called sex-linked inheritance or X-linked inheritance. Let's call it sex or X-linked inheritance. And you see why it's called that, right? Because these traits go with the sex chromosomes, the X chromosomes there, okay? Sex or X-linked inheritance. And here are some examples of those. All right, so that pretty much brings us to the end. Let's quickly take a look. Yeah, so what we are seeing here is, even though we see how genetics work, but they are not the entire story. an equally important part of the story is also your environment, right? So for example, if you have the gene, the genotype for being very tall, but you never got first-class protein, good nutrition in your life, or you were confined in a small room or whatever, so you will not attain that genetically predetermined height because of your environment. So environmental factors also have to basically taken into consideration when you look uh, at genetics. So like it says, alleles with the risk of cancer development. You might have the cancer-prone genes, but if you never smoke, follow a healthy diet and exercise, they might never express themselves, you see? So genes are not necessarily destiny, at least not 100%. So what would be the results in terms of the genotype and phenotype if one parent for heterozygous for the widow's peak and the other parent for homozygous? So we have to make the cross and see what happens. How does codominant differ from incomplete dominance? Codominance when two traits are equally expressed, like AB, both are expressed, AB blood type. Incomplete dominance is when two traits intermix and an intermediate phenotype comes out, like dark skin and light skin, you get brown skin. That's incomplete dominance. If a woman who is homozygous for color blindness has children with a man having normal vision, what would be the potential genotypes and phenotypes of the offspring? Make the cross, make the tic-tac-toe, and figure out what it's going to be. How would nutrition affect growth where hereditary factors were equal? So like I said, you might have the genes for growing tall, but if you don't get first-class nutrition, protein, all that, or not enough exercise, you will not attain your genetically determined height. All right, so that is it. The end of our semester, at least the lecture section, okay? So it was wonderful interacting with you all and seeing your progress throughout. Hopefully the feelings are mutual. And so again, you have your study guides, everything sent out. So please use those to the best of your advantage. Make sure you do your case studies and turn them in before the deadline, as well as your lecture quizzes on the Connect website. They are there as well. Any questions, suggestions, feedback, please do not hesitate to let me know. And with that, have a wonderful rest of the weekend, wonderful rest of the week, and hopefully I'll be checking in on you guys virtually, at least with your exams and everything. All right, so have a great one. Bye-bye.